So again, you can see here the cell upon insertion expand the apical domain, and later on you have formation of the cilia from the apex of the cell. But interestingly, if you, for example, zoom out and look at the bigger chunk of the tissue, you will actually see that in principle you have hundreds of these multi-seated cells progenitors in green, and you have also hundreds of these vertices in the tissue, which poses quite interesting question. How does the cell actually determine in which vertex to insert? So it's actually kind of linking to our previous session. How does the cell perceive the environment? How does the cell decide, okay, we have here 3 vertices, but I'm going to select this vertex versus the other one. So partial answer to this question comes from dynamic imaging. So here I'm going to show you a movie where migrating cell progenitor in green. Basically, it will appear in here as it moves towards the top of the tissue. And just to orient you, I will also put here the positions where you have, for example, 3 tricellular junctions. So as I played this movie, hopefully you can appreciate that these multi-seated cells, they're making these very dynamic protrusions, finger-like looking protrusions called Philopodia. So you can see here that these Philopodia are growing from the leading edge of the cell. And previously, these Philopodia have been associated with filling the passive cellular environment, for example, extracellular matrix. And then we thought, okay, maybe in our system, this Philopodia could also sense the active, so cellular environment of the tissue. So how could it work? How could it work? So to test this possibility, we first decided to quantify the dynamics of Philopodia. So to this aim, we first sectioned the movie along the vertical axis, which gave us this kind of representation where you have the body of the cell, and you can see this growing Philopodia, and you can also appreciate the position of these tricellular junctions. And in the second step, we basically quantify the position of the Philopodia within the leading edge of the cell. So if this is a schematic representation of the cell, you have this Philopodia growing here. We basically automatically segment the leading edge of the cell and score the position of Philopodia within this leading edge of the cell. So this gives us something what we call Philopodia index, represented in a way that we have this leading edge of the cell starting from the beginning until the end, and then whenever we have Philopodia present within the leading edge, we mark it with this magenta color. So if you do this exercise for the entire length of the movie, you get something what we call this Philopodia map. So how to read it? And again, here on the x-axis, we have this position of the leading edge of the cell. On the y-axis, we have time. And here we also mark the positions of the left and the right tricellular junction. So these 2 lines. So this is basically the tricellular junctions which the cell is interacting with. So when you look at many of these movies, it's kind of apparent that this Philopodia are kind of growing, overlying the positions of the tricellular junctions. So you see here that, okay, there's a lot of Philopodia underneath this left tricellular junctions and also underneath this right tricellular junction. So what is going on? Then we wanted to know, okay, do cells just make this Philopodia just pointing towards these tricellular junctions, or actually do this Philopodia actively binding these tricellular junctions? Yeah? So to test this possibility, to address this question, we express this protein called LSR. So this is just a protein that kind of hangs down from the tricellular junctions down into the tissue. So it doesn't matter what kind of protein it is. It's basically like a cable that hangs down into the tissue. So now the next step, we want to basically see, okay, are there direct interactions or not? So simply we image this process. So again, you can already see these cables kind of hanging down from the tricellular junctions, and then at some point, when I play the movie, you start seeing these cells moving from the bottom and making these Philopodia. And it was very clear that these Philopodia are actively binding these cables, actively connecting with these cables at these vertices. So at this stage of the project, we still don't really have any clue what these Philopodia are doing, in the context of developing mucociliary epithelium, until we start looking kind of quite carefully at these interactions between Philopodia and these tricellular junctions. So often when we do so, we see that Philopodia are actively pulling at these vertices. So basically they grab the vertex, these Philopodia grab the vertex and basically yank on this vertex, pull it down. So then we thought, okay, maybe these pulling forces somehow could explain the function of Philopodia in this process that we study. So if this is still not clear what I'm talking about, just bear with me for a second and just look at these schematics. So for example, if you consider for a second, you have two elements like a soft rubber ball and you have a hard or non-compliant element such as hard kettlebell, and then you imagine that you connect the ropes to these two elements, and you think about that you just pull down, yank on this rope. So you as a human, you will be able to determine, okay, this is a soft element and this is the rigid element. Yeah. So now the question is, can cells do the same? Can cell actually pull on these different vertices and can determine, okay, this is a soft vertex and this is the rigid vertex? And also the second question is why the cell would do so. Is it any beneficial for the cell to read this environment in this way? So as often in our lab, when we don't know how the things work, we basically set up a theoretical modeling, and this is exactly what we did in that case. So here we wanted to model what's the function of the pulling forces in basically reading the environment of the tissue. So the model consists of 3 steps. In the first step, we basically describe the mechanics of the tissue using standard vertex-based model. So again, this is a classical approach where we consider tissue as a network of connected vertices, and each vertex has a certain energy which is given by multiple parameters. And for example, this parameter can be, for example, the rigidity of the cell and also the tension of the junctions. So these bonds, how strong they are connected to each other. In the second step of the model, we applied the pulling forces to the vertices. So basically, this is to mimic these pulling forces that the Philopodia are exerted on the vertices. So in the second step, basically, if we start with the vertex here, and then we apply the pulling force F, if you know exactly the amount of the pulling force, and we know the displacement of the vertex, we can estimate something what we call vertex stiffness. And if you do this exercise for all of the vertices in the tissue, we get something what we call vertex map, vertex stiffness map. So basically you can see here that some of the vertices are stiffer and some of the vertices are less stiff. Yeah? And in the last step of the model, we basically measure the probability of the cell expanding the vertex. So here, if we have our tissue in which we measure the stiffness of the vertices, in this step we basically introduce the cells underneath the vertex, and we measure the probability of expansion of the vertex. So what was interesting, coming out of the model, that the model predicted 2 things. So one thing is that cells will insert the stiffer vertices, so the stiffer the vertex is, the easier it is for the cell to insert. And the second prediction was that the cells really prefer to insert at these higher order vertices, meaning 4-fold or higher fold vertices. So 4-fold vertex, the vertex where it's connected to, let's say, 4 bonds, whereas the 3-way vertex is the vertex that's connected to 3 bonds. And without going much into details, the reason for this discrepancy between the 3-fold vertex and the 4-fold vertex is simply because in the case of the 4-fold vertex, you have basically additional pulling forces. They basically help the vertex to be expanded, to be opened. So while generally the model was quite informative and predicted quite well what we see in the experimental data, there was still one thing was a little bit puzzling to us. Because, for example, the model predicted that the cells prefer to insert at the higher order vertices. Yeah? And indeed, if we look at the later stages, we see that these multicellular cells here are marked in this magenta color that indeed prefer to open at the higher order vertices. But if we think about it, when we look at our data, there are basically no 4-fold vertices present in the tissue. So if you look at the initial stage of the tissue, there are mostly 3-fold vertices. So what is going on? How do you get 4-fold vertices out of the 3-fold vertices in this system? So if you think about this question, there's only one kind of possibility to get the 4-fold vertex out of the 3-fold vertex. Namely, this junction that connects 2 3-fold vertices has to shrink. But now the question is, okay, who is shrinking this vertex? Who is shrinking this bond? is this shrinking of the junction happening within the tissue, or actually these multi-seated cells are shrinking this junction. So to test that, again, we look at the processes where we see the formation of the four-fold vertex out of the three-fold vertex. So here we're looking at the top of the tissue, and here we see, for example, that these two bonds are going to shrink, contract, and they will form this higher order vertex. On the right-hand side, you actually see the corresponding position of the multi-seated cell. So let me just play the movie. It will be maybe a little bit clearer. So as I play the movie, you see that these two junctions are shrinking, and then it is kind of clear that these multi-seated cells correspond to the position of the shrinking junction. So it looks like that the multi-seated cells are in a close proximity to the shrinking junction. And maybe it's a little bit clearer if I look at the same process from the orthogonal perspective. So we look now at the same process kind of looking from the side. So if you look now at the junction that is shrinking, so this one, and you also look at this multi-seated cells here in this pink color, you see that these multi-seated cells are actively probing the shrinking junction. Actually, you can see this Philopodia almost binding these vertices overlying the cell. And then if you quantify this, dynamics of this process, it will look like this. So traditionally, when you think about shrinking of the junction, we have to think about myosin, because myosin is this motor protein in here that pretty much like in our muscle, it will contract and bring the actin filaments together. It will shrink the junction. So if you, for example, measure the intensity of myosin along the shrinking junction, this is what we get. So first of all, if you now look at the length of the junction here, this purple color, you see that starting from kind of time point 20 minutes, the junction shrinks. Yeah, so it goes to almost 4 micrometers. But if you look at the intensity of myosin, so again, this thing along this junction as a proxy of the tension along this junction, actually the tension of the junction is pretty stable and only increases at the later stages. So this basically tells you that, okay, myosin is probably not really involved in shrinking this junction. But then what is happening with this multi-seated cell? So we can also quantify the position of the multi-seated cell looking at this actin intensity. So again, if I measure the intensity of actin, as the cell is approaching the overlying junction, it will look like this. So this is this green line. So it was very interesting that we can see that beginning of the shortening of the junction very nicely corresponds to the time point where this initiation of the actin intensity increases. So meaning that as the cell is kind of coming closer to the junction starts to shrink. And most interestingly was the fact that exactly at the time point where we start seeing the beginning of the junction shrinking, so basically kind of minute 21, so this is in here, this is the first time point where we can see that the Philopodia are grabbing these two vertices. So in that case, meaning that Philopodia not only can probe the vertices, but they can also grab two vertices and bring them together. So then what will happen if we, for example, perturb Philopodia activity? What if we, for example, make the cells without Philopodia? So this is also what we did. So for example, if I play now this movie in which we perturb the Philopodia activity in some cells. So for example, whenever you're going to see a cell that expresses a pink color in the nucleus, this is the cell in which we perturb Philopodia activity. So you can see here that some of the cells actually, like this one, it manages to go to the top of the tissue, but at some point just disappears. So it never manages to insert into the vertex and open the apical domain. So basically it completely lost the perception of the environment. So with that, let me just summarize. So today, I was trying to basically answer the question, how do cells sense this cellular environment? And it looks like that the cells are actively probing the environment using the Philopodia. And they're using this Philopodia to actually actively grab the vertices and pull on the vertices to estimate how stiff the vertex is. However, in most of the cases, the stiffness of the vertex is not high enough. There is not enough of the kind of threshold that the cell will move over and will be able to insert. But in that case, the cell is able to basically remodel its environment by bringing this, by basically collapsing one junction and forming a four-way vertex out of the three-way vertex, which is much more susceptible for the cell to insert into the tissue. So this is very cool because it was, one of the first representation that shows that, that the cell can not only read the environment, but can also actively remodel the environment by itself. So with that, I would like to thank my entire lab, and I will be happy to take questions. Thank you very much.
Fantastic.
Was it on time?
Thank you. Yes, wonderful, wonderful. I didn't even have to stand up. So perfect. Thank you. We have time for some questions. Yes, I can see Kasia. I also have a question. So I want one.
Hi, thank you for the very interesting presentation and the beautiful imaging. My question is, I guess, I know more about the probing function of the Philopodia more than the actual pulling. So I'm just wondering if this mechanism that you described is just specific to the places where the cilia will later grow on, or can it be at all transferred to, I don't know, cancer cells or immune system or anywhere else?
Yeah, it's a great question. It's actually a very general phenomenon, I would say, because, I mean, of course, I didn't really get much into detail, but on the tip of this Philopodia, you have this protein called LSR. So this protein LSR is also present within the tricellular junction. So you basically have a situation when you have the protein within the tissue kind of hanging down, and also have the same protein at the tip of the Philopodia. So when I talk about this binding or interactions between Philopodia with tricellular junctions, actually this binding is happening through these LSR interactions. And what is very interesting, that LSR is implemented, as a marker of multiple different types of cancers. Yeah. So, for example, if you overexpress or downregulate LSR, it completely changes the, metastatic dynamics of different cancers and so on. But it's not only, let's say, in the pathological conditions and so on, but this kind of probing by Philopodia, this is also present in other different, cell types during development. So, for example, in the olfactory system. Yeah, or in the kidney and so on. So there are multiple different examples where Philopodia are present. It just, people haven't looked detailed enough because, for many of these aspects, you need to have, quite fast, spatial temporal resolution imaging, and, actually take time to look into fine details. So I hope that, we can go into this direction a little bit more in the future.
Thank you very much. I have a question.
Yeah.
To follow up a bit on the LSR protein, I was actually wondering, what if you overexpress certain proteins in these cells? Will they produce more?
Yeah, this is actually what we did. So if we overexpress this protein, specifically LSR, in these multicellular cells, then the cell is basically covered with Philopodia. Yeah, but the question is, okay, does it help the cell to have more Philopodia in kind of finding, the destination? Yeah. Is it beneficial? But the answer was not. So at the end, what turned out to be, the case is that it's not about, how many Philopodia you have it, but how long the Philopodia is binding the junction. Yeah. So basically about the timing of the connection.
Fantastic. Thank you very much.
Thank you so much.
Thank you.
Cheers.
I'll introduce our next speaker. Our next speaker is Magdalena Laska. Magdalena is an associate professor and the leader of an AMBER group at Aarhus University, and AMBER stands for autoimmune modulated by endogenous retroviruses. I'm very excited about this presentation because I actually do like viruses and worked with them and now have to read a lot about them. Yes, the research group is at the Department of Molecular Biology and Genetics. She obtained her PhD from Aarhus University and Cold Spring Harbor Lab in the US. She is a very accomplished scientist and entrepreneur with some patents up her sleeve and many, many publications in great journals. So welcome, Magdalena, and thank you very much.
It's very loud, isn't it? No. Thank you for the great introduction. So, as you heard, I will be talking about viruses, and I know most of you are tired to hear the word virus, but I'm going to talk about different viruses. I'm going to talk about ancient viruses, which are actually part of all of us. You have them, I have them, you have them. So we are going to talk why they are there, how they got there, and what we can do, or shall we do something about them. So the title of my talk is Ancient Viruses Within Us, a journey from basic science to new drug candidates. So this is a short agenda for my talk. First, I will give a personal statement, which basically already was provided. Then I will tell you what is it all about and what are those human endogenous retroviruses, why we are all partially viral. Is it good or bad? Sounds bad to me, but maybe it's not. How they regulate our body, our immune system. Are there any clinical strategies, drugs, which target those viruses? Shall we target them? And then I tell you a story or a project spinning from my laboratory where actually those enemies or those foes turn out to be our friends. How is that even possible? So, as mentioned before, I'm an associate professor at the Aarhus University. If you wonder where is Aarhus, is the second city in Denmark, the second largest city. It's very beautiful. I encourage you to come and visit. We have really beautiful campus. So my laboratory works with viruses, mainly endogenous viruses, meaning the one within us. And what we do, we are trying to study, as I like to call it, a paradigm shifting hypothesis about the interaction between viruses and the human immune system in autoimmune diseases and cancer. Okay, so human endogenous retroviruses. I told you that we are all partially viral. Actually, if you look at the content of our genome, so the protein coding genes, the one we know, they consist very tiny percent of all the genome. And those human endogenous retroviruses, they're quite large portion, up to 8% of our genome. But how on earth they got there? What happened? So we have to go back million of years ago. So what happened is that a virus, the one being in the environment, the one you heard about, HIV, SARS, and so on, infected our ancestors. But it infected a very specific cells, germline cells, meaning egg and sperm. And what happened is that the virus liked the host so much that it decided to integrate into its DNA, become a part of DNA. And then the virus started to be transmitted from one generation to another generation, into another generation, until it became a part of our DNA. So once again, this is Those viruses are transmitted vertically, meaning from the offspring, from the mother to the offspring, and so on. So the virus infected germline cells, and then it got fixed, endogenized into our DNA. So we are all part viral, like it or not. What happened during this process? It took millions of years. The genes which code for different proteins building the virus, they got a lot of mutations, deletions, or other changes, making them usually inactive. But some of them, very small percentage, about 50 in our genome, they are actually still quite active. They are able to produce functional proteins, which then later can react with our body, with our immune system. So, are those viruses which are the part of our DNA bad or good? Well, I will first stand on this side, which shows the bad. Well, yes, in some ways they can contribute to many different diseases, which we like to call autoimmune diseases. Autoimmune meaning that your body starts attacking your own cells and tissue because it cannot recognize what is foreign, what is the pathogen, and what are your own cells. And those protein expressed by those endogenous retroviruses has been linked to many autoimmune disorders such as rheumatoid arthritis, diabetes, multiple sclerosis, and so on. And how actually they can contribute to the disease, right? How those protein which comes from those endogenous viruses can cause the disease. Well, they can start dysregulating your immune system, meaning that they start activating your cells to produce specific pro-inflammatory cytokines or chemokines. They can activate other genes and so on. On the other side of the scale, we have a beneficial function. And this is where the emoji with the focus is. This is where my research is. So there are two hypotheses. One is that, yes, those sequences are not just a junk DNA, they actually do play functions. Those viruses can produce proteins, can be reactivated during certain diseases like autoimmune diseases. But on the other hand, you might ask, okay, they were kept in our genome for millions of years. Maybe they do have actually some important function as well, some beneficial function or some physiological function. And yes, that is true. Some of them play beneficial function. For example, there is a specific endogenous virus which is necessary to form a syncytial trophoblast. This is a part of the human placenta. So during the pregnancy, actually some of those viruses called syncytins, they are necessary to build a functional human placenta. So, okay, so we have them. We know that they can be both a little bit good or bad. And I will tell you sort of two stories why, one story showing what that the harmful function of them, and the other what are the beneficial function of them. So the best story, or the best well-known story, is about contribution of those viruses is multiple sclerosis. Maybe you heard about it, maybe you have someone in the family who is affected. So multiple sclerosis is a very unpredictable, often debilitating disease, much more frequent in female than in male. And this disease involves immune-mediated processes in which there is abnormal reaction from our immune system, which targets our central nervous system. So, as you can see, this is a nerve cell in a healthy individual, and it's sort of covered with this, I would call it protective blanket, myelin, it's called. But what happens in the multiple sclerosis is that this layer, this protective layer, myelin, is damaged, right? So this is the cause or the consequence of the multiple sclerosis. So how on earth endogenous virus could be one of the causative factors in multiple sclerosis? So this is the same cell, and you can see there is this myelin which is very, very damaged. So one sort of hypothesis, or which was later proven, is that you have endogenous virus which produces a protein, here shown in green. And what happens is that there are some receptors, maybe you heard toll-like receptor 4, on cells which are called microglia. And what happens is that if the viral protein binds to those receptors, those microglia cells start producing pro-inflammatory molecules, pro-inflammatory cytokines. And already, as the name suggests, pro-inflammatory, that's not good. You want to have something anti-inflammatory, fighting the inflammation. The other thing is that those cells, usually, they clean all this debris. They scavenge the debris. And when the viral proteins bind to toll-like receptors, they cannot clean up this debris, which is extremely important process to actually rebuild this myelin. So this is very bad. So when you think about, okay, what we can do about it and to help patients with multiple sclerosis, maybe we can simply block somehow the production of this viral-derived protein. Are there any company pursuing this approach, or is it gonna work? Yes. So there is a Swiss-French company called Geneuro, and they developed an antibody which will bind to this viral protein, and it will block it, right? It will inactivate it and it will block it. Does it work? Well, they tested this antibody is called temelimab. It was tested in multiple sclerosis, and they are now actually in already phase 3 clinical study, so very late, very large studies, almost ready to be on the market. The same antibody or the same drug was also tested in type 1 diabetes. Now they are in phase 2B of their clinical study, and also the same drug can be used for other disorders like inflammatory psychosis and so on. So I showed you that we do have some viral sequences which in specific conditions, autoimmune conditions, can produce proteins which can be harmful to our body. But there are some clinical strategies to deal with it. Now I'm going to tell you something different. Remember the scale, sort of detrimental versus beneficial function. So I told you that in my laboratory, we are focusing on, I would say, advantages or beneficial functions of those viruses. And we actually discovered them, like all great discoveries, by accident. So we work with the disease called lupus, or systemic lupus erythematosus, quite often. And this is also, again, autoimmune disease, meaning your body attacks its own tissues, its own cells. And the quite well-known characteristic of the lupus is so-called butterfly rash. So the patients have here on their here on the area a very pronounced rash. It's also called a wolf disease. And we ask a very simple question. Okay, we take a patient which suffer from lupus and healthy controls and try to measure the expression of those viral-derived proteins in their organism, in their blood. And we found that there was protein coming from one specific virus, which was very highly overexpressed, meaning there was more in SLE patients or lupus patients than in control. And our logical / non-logical thinking was like, if there is more of something, that's probably bad, right? We tend to think that if something is overexpressed in the disease, that's probably a factor, a causative factor in the disease. But it actually turned out to be opposite. We did a lot of studies correlating the expression of this specific virus to the clinical manifestations, how well the patients are doing. And everything was always reversed, meaning the more virus, the better patients were actually doing. So we started to hypothesize that maybe the virus or the viral-derived sequences are there to benefit the patients, to help them fight this disease. So what we did? So we looked very, very, very closely using different molecular biology tools into this protein, which is expressed from this virus, and we were able to sort of identify where this activity is located, to which domain, and this domain is known immunosuppressive domain. And what we did is we look at the sequence, right, and we design, based on this sequence, a synthetic peptide, right? So synthetic peptide, which sequence like a small molecule, which sequence corresponds to exactly the same sequence, right? And those peptides, they only contain naturally occurring amino acids. There are no artificial amino acids. And we screen them. We screen them for the solubility, cytotoxicity, and functionality. We also now know the mechanism of action, which I'm not gonna talk about. But what happened is that we end up with the vial of peptide, which looks like this. I brought it for demonstration. It's basically a little glass with some white powder inside. And we try to validate it. We try to test it. Okay, how do we test it in the cell culture, in the patient materials, but also in animal models? And remember, this all comes from the virus, which is inside us, right? So what we found out that when you treat the cells with the peptides, they inhibit the production of those pro-inflammatory molecules, which are often expressed in those patients, right? One of those molecules is, for example, interleukin-6, right? So treatment with the peptide inhibits production of those molecules. What we do next is we actually do take the cells directly from the patients. We call it ex vivo analysis, right? And remember, those patients are very sick, right? They are quite sick, so they have a lot of those inflammation molecules already expressed in their body. And we treat their cells with the peptide and see if we can inhibit the production or of those molecules. And yes, we can do that. But the real test comes usually from the animal models. And we tested our peptides in a variety of animal models. We tested in the multiple sclerosis, inflammatory bowel disease, rheumatoid arthritis, autoimmune arthritis, and also in type 1 diabetes as a collaborative project with Novo Nordisk. So this is example from, for example, rheumatoid arthritis. So on the y-axis, you have sort of showing you how sick are the animals. This is a disease score, right? So we induce the disease in the animals. If they are not treated, they're just becoming very, very sick. If we treat them with our peptides, they are doing much, much better. They are much less sick. We later optimized the peptide that actually is able to inhibit 90% of arthritis. So very good. The same is true for multiple sclerosis. This is again the disease score. We induce multiple sclerosis in animals. If they are not treated, they are very sick. If we treat them with, as I called, prototype peptide, they are doing much better. Later, we optimized the peptide that, in the case of multiple sclerosis, can inhibit 85% of the disease. And remember, I showed you this nerve cells with this myelin, right? So this is multiple sclerosis, right? So if you don't treat the animals, what we are looking is this more bluish layer. The myelin is gone, but when we treat with the peptides, the myelin is actually preserved, meaning the animals are doing much better. They also do much better. The level of all those markers which are associated with disease, pro-inflammatory markers, are greatly reduced when we treat them with the peptides. So those viral-derived peptides actually can be used as medicines, right? They are simple molecules. They are quite cheap to produce. They are contain only human-derived amino acid, right? And they don't show any surprises in their metabolics or kinetics. So to summarize, what are my next steps, or in my research group, or what I'm also hoping to achieve via networking here, is that we already optimized the peptides designed to increase their potency, so they work much more efficiently. We are testing the peptides in different diseases, like rare diseases, of orphan diseases. Because of the mechanism of action, which I didn't talk about, they have application to different conditions based on their target. We are trying to generate new IPR on new peptides and working on our commercialization strategy. Thank you.
Thank you very much, Magdalena. Now it's time for questions. Yes, Krzysztof.
Hi, thank you for the talk. I have two questions, actually. Okay. So first one, well, as it usually comes with a new form of treatment, do you see any side effects in those mice? Because of course the interleukin and like inflammatory pathways are also important for some countries. Yeah, so that's first. And the second one, is like every person, like how that candidate go that every person has exactly the same viruses then? Like, because do we come from exactly like one mother? No. So like how did it propagate exactly to whole population?
So I will start with answering on the second question. It's because they integrated so many years ago, right? It's millions, 100 millions ago. The research, the youngest, so to speak, infection and integration was approximately 8 million years ago. But we do have polymorphism in them. That's possible, right? But yes, it's because they integrated so long, so we actually have almost the same genetic makeup if it comes to the viral sequences. But they do have, you have to remember that they are, most of them are inactive. There's only very few which have completed so-called open reading frames, meaning they can produce proteins which can have effect on your immune system. And answering the first question is that we do not see any side effect in the mice we treat, but always there is some concern with the peptide molecules and using them as a medicines. That's usually due to their very short half-life, but there are also mechanisms to increase or to make them more stable. There are different modifications. There are options to have it on skin patches, for example, where they will be systematically released over a long period of time. But for now, we do not see side effects, and I believe it is linked to the fact that one of the facts could be that they contains naturally occurring amino acids. I'm not saying that not natural amino acids are bad, I know, but so I would just say that for now we do not see any side effects. But we are doing more for toxicology and all those studies at the moment.
Thank you. I have one question about the viruses that are actually harmful.
Yeah.
They produce something that is harmful for us. What's the field of genome editing saying about editing it?
Well, it's very difficult because you have to think that there's so many of them and they're extremely, extremely similar to each other. They are so homologous, right? So the difference between them are very, very tiny, right? So I believe that it will be quite difficult to, even if you look at the, for example, if you would like to study polymorphism or something, right? It's very difficult because the sequences are so homologous, right? So usually you actually place your markers a little bit outside the viral region, right? To be able to localize where exactly is your association coming from. Because you can also study it genetically, right? And think about them as just genes. Yeah?
So I have a question. So how do you test the effects for the stability?
Could you please repeat the question?
The question was, how do I test for the stability?
Yeah.
Well, that is a good question. We do not, I would say that, in our laboratory, this is not what we do for stability, right? This is where we try to develop assays with the collaboration of the company, which can measure then, how fast the peptides are removed and so on. So stability but you can also think about the stability a little bit from the functional point of view, because it depends how frequently you inject it, right? So you can also think that, because in some of the studies we started with daily injections, right? And we see great effect. But daily injections for most of the diseases is just no go. It's too much, right? You want to have it as a one per week, right? But in the studies, for example, like we did with the diabetes, right? We treat the animals and then we withdraw the treatment, right? And the effect lasts for quite a long period of time, right? So I honestly believe that maybe they could bind to some serum-related proteins in the blood as a carrier, and that would actually maybe also help them to be more stable.
Yeah, also in the case of the degradation, because one thing is just removing them, and the second question is degradation.
Yeah.
They can be immediately basically degraded.
Yeah.
Within a second after you inject them.
Yeah, that's true.
So that was my question.
No, but there are assays where you can actually measure the speed of
So my question was how you measure and if you measure.
Yes, we do, but this is done not in our laboratory, but as a CRO service. Yeah.
A very, very short question and brief answer.
So I was wondering, what is known about the secretion of these virally encoded proteins? You said that upon the damage of the myelin sheaths, you saw this burst or release of the protein. Do you know what is triggering that, and what type of cells are capable of secreting?
Yes, we know a little bit. Well, you could also think that normally those sequences are silent, right? And that also has to do with the methylation of, so there are some mechanisms which would just start the transcriptional activation, which then leads to the secretion of those proteins. For example, in multiple sclerosis, we see that CD4 plus and CD8A plus positive cells are the one starting to produce the proteins, which we can measure the level of it. But normally, in most cases, they are just silenced by the methylation. So actually, methylation has quite a lot to do with removing the methylation marks and start the activation of the sequences. Thank you.
Thank you very much. Yes. And we will go very shortly to the talk delivered by our third speaker of this session today. Actually, the aim of this meeting was to gather professionals from different fields, but also across the stages they are in their career. That's why we have PhD students, we have PIs, we have leaders, we have developers, and so on. And Kasia, I believe, is our youngest presenter today, so please give her a especially warm welcome when the time comes. She's currently a PhD candidate in the Life Science Zurich Graduate School. She graduated from Jagiellonian University and is very interested in disease-related and protein stability, actually. And during her work, she got familiarized with the concept of structural biology and decided to continue with that. She was invited by EMBO to an interview to get their fellowship, but she said no to it, to be in the group where she is right now. So I think she really knows what she wants. And today she will tell us about the channels and the structure of different channels. So give a very warm welcome to Katarzyna.
Thank you very much, and can you hear me well? Okay, so welcome everyone. Let me accommodate myself somehow here. So welcome everyone, and thank you Agnieszka for the introduction, and thank you for the invitation. And I will talk to you today about a pretty mysterious family of ion channels and their shapes, but let me first start with a bit more general background. So here you can see a schematic picture of a cell, and essentially all the processes in a cell that you can imagine rely on the proteins, whereas proteins, to fulfill their functions, relies on their 3D structures. 3D structures that are determined by the amino acid sequences, so polypeptide chains that form those proteins. And I am a part of the structural biology group, and in our works we are mainly focused on determining such 3D structures of the proteins, simply in order to get the better insight into their function and their mechanisms of action. And of course, I'm not studying the structures of all the proteins, but I'm focused on the small family of calm ion channels. And calm stands for calcium homeostasis modulators, and it is a family that consists of 6 members, 6 proteins, simply named from 1 to 6. And when I started my project, no structure of any of those family members has been available. And also our knowledge about their functions is pretty limited. So our knowledge about the functions of calm proteins is restricted to 2 of the family members, which are calm1 and calm3. And those 2 proteins are known to pair together to form the channels in the taste buds. And what happens where they participate in the taste perception. And what happens in taste buds is that upon the detection of taste buds, the signaling cascade is activated in those cells that eventually results in the opening of calm1-3 ion channels that then release ATP that serves as a neurotransmitter for the gustatory nerves. And this was a pretty interesting finding, actually very interesting finding, because ion channels are usually known to conduct single charged atoms like sodium, potassium, or chloride. But here we have the channels that are able to accommodate huge molecules that ATP is. And this poses a question of how those channels can accommodate such huge molecules. And to figure this out, to understand it, we decided to attempt to determine the structures of those calm channels. And when it comes to the structural biology, structural determination, one actually needs quite huge amounts of proteins. And this is done by the artificial production of the protein of interest into various expression systems. And in my project, I used the mammalian cells, which I basically forced to produce the protein of interest for me. And then the next step was the extraction of those proteins. And I also had to somehow check their biochemical state, their biochemical stability, to make sure that we can work with these proteins, that they are properly folded, and we will be really able to obtain the structures from them. And as you can imagine, we the most wanted to determine the structure of calm1 or calm3 proteins that have the function assigned. However, in the size exclusion profiles, size exclusion is a type of chromatography that allows us to separate proteins, to isolate them, to purify them based on their size. And it works in a way that the bigger the protein, the earlier it elutes on the column. But actually there is also something that always elutes at the very beginning of the columns, which are huge aggregates of proteins. And this is something that we really want to avoid because we cannot work with protein aggregates because those are usually misfolded proteins. But this is what we got. This is what we got for CAN1 and CAN3 proteins. However, we were more lucky with the other members of the family, out of which the best one, the one that performed the best, was CAN4. That gave us a huge peak, eluting at the volume that indicated stable protein. And this huge peak indicated that we can purify, we can obtain large amount of this protein for our structural studies. So for that reason, we focused on CAN4 from then on. But before starting to work with that protein, we actually wanted to put it in some physiological context. But the problem was that, like I already mentioned, we didn't know the function of that protein. And even worse, we didn't even know where it is expressed. So in what organ, tissue, or cell it can play a role. The only hint about this protein that we had was the data from this database that showed high mRNA levels of CAN4 in the placenta. And mRNA is simply a template at which the proteins are produced. So one can assume, it's not always the case, but sometimes it is that the more of the mRNA in the particular organ or tissue, then the more protein is there, and it might play an important role in this organ. So because of this finding, we started the collaboration with the group of Professor Christian Albrecht that studies placental transport, and we asked them to determine mRNA levels of all CANs in the placental tissue. And here is what they found. So they found that, they indeed found high mRNA levels for CAN4, but also for CAN2 and for CAN6, but not for all three other CANs. And even more interestingly, they had found that mRNA levels for those three CANs increase upon the differentiation of the placental cells into a structure, actually the structure tissue that you could already hear in the previous presentation, so syncytiotrophoblasts, that is responsible for the transport across the placenta and exchange of the substances. So you can imagine that this was a very interesting finding for us, and from then on we decided to focus on those three homologs, on those three paralogs, so CAN2, CAN4, and CAN6, and to determine the structure of all three. And upon the standard two-step purification, we obtained pure samples of our proteins, CAN2, CAN4, and CAN6. And already at the final step of the purification, which was size exclusion chromatography, we could see some interesting features of those proteins. So one CAN is actually a very small protein. It's only 30 kilodaltons, whereas those proteins eluted at the columns, at the volumes, elution volumes that would indicate huge complexes. So we basically suspected that they probably oligomerized to form bigger complexes. And there is a fantastic method perfectly suitable for determining structures of huge complexes, which is Cryo-Electron Microscopy. And this was our method of choice to approach the structures of those three proteins. And now I would like to tell you a bit about sample preparation for cryo-EM, as this is really a crucial step to get the sample of a good quality. So in this method, we use very teeny tiny metal grids at which we apply our protein sample. Then the next step is the removal of the excess of the solvent, because we really want the thin layer, the thin film of the protein sample at this grid. And the final step is very rapid plunging of this grid into liquid ethane. And as a result of it, we get our proteins, hopefully at random orientations, frozen in a very specific type of ice called vitreous ice. And this ice differs from the regular ice in a way that it is devoid of the ice crystals. And having such grid prepared, we insert this tiny grid into this huge electron microscope that you can see here on the right. And then we simply sit in front of those computers that you can see on the left for 6, 10, or 12 hours, sometimes 14 hours, trying to set up the data collection that in case of my proteins lasted overnight. And the result of such data collection are so-called micrographs. Here you can see the micrograph that was collected for CAN2 protein. And micrograph is simply a picture of those proteins frozen in those vitreous eyes at the huge magnification. And we collect thousands of such micrographs. And here is where the most exciting part of cryo-EM analysis starts, which is data collection, which is data processing. And the first step in the data processing is called picking, in which software recognizes protein particles on those micrographs that here in case of CAN2 appears as the ring. So the software picks all of them and then classifies them into so-called 2D classes. So it basically groups them into different classes, different groups based on their orientation. So based on the angle at which we can see them. So top views, side views, or tilted views. And having that we have good 2D classes, we can approach so-called 3D reconstruction to obtain the map, 3D electron density map, that would reflect the shape of our protein. And as you can see, we tried this with CAN2. Nevertheless, for CAN2 protein, we weren't really able to obtain the map of resolution high enough that would then allow us to build the atomic models of CAN2. And this was due to the preferred orientation, so sometimes it happens, and it happened for us with CAN2 that it displayed this preferred orientation on the grid, so it was mainly present as we could mainly see the top views, and we didn't have enough side views or tilted views to build the sides of this map. Nevertheless, we were more lucky with two other proteins that we studied, so CAN4 and CAN6. And for those two proteins, we obtained beautiful cryo-electron density maps at the resolution that allowed us to then place in those densities the amino acid sequences that are known to build those proteins. So let me now guide you through all those maps and those structures, and to tell you what conclusions we could make once having those structures. So let me start with CAN4. And for CAN4, we obtained two electron density maps. And what you can see here, this doesn't really represent the one CAN4 protein. This represents the complexes that CAN4 proteins forms. So here you can see the map from the side, and here you can see it from the top. And those CANs, as we could see from those maps, they oligomerize, so they form huge channel-like complexes that contain of either 10 CAN4 proteins or 11 CAN4 proteins. And also we noticed very weird thing, which was that those channels dimerized through the intracellular parts. However, I wouldn't like to focus on this part because we believe that this is simply the artifact from the overexpression of the protein, and we believe that it doesn't really have the physiological relevance. So let me just focus on the structures of those channels that CAN4 forms. So here you can see the atomic models that we built using those maps. And like I mentioned, CAN4 proteins oligomerize to form oligomerize that in a structure that in a way that results eventually in the formation of the huge pore in the middle. And believe me that here I indicated the diameters of those pores, which is 25 angstrom for the decamer of the CAN4 and 30 angstrom for the undecamer. And those are really exceptionally huge pores because the other proteins, connexins, that are known to form that are known to form channels of large pores, in their case, the pores that are considered large have the diameter of around 15 angstroms. And here we have the diameter of even 30 angstroms. And here you can see the space filling models of those two channels, seen from the top. And here what is placed inside is the model of ATP. So you can probably appreciate already that this oligomerization is a way in which that allows those channels to conduct such huge molecules. What comes to the structure of one single protomere of CAN4, so we could see that CAN4 displays a new fold. So that means that it adopts the structure that hasn't been yet described for any other protein, what indicated that it probably works in a way that hasn't been yet described for any other proteins. And very briefly, I would like to tell you what is the structure of such CAN4. So it is formed by 4 transmembrane helices, out of which the most important for one, the most interesting for one, is transmembrane helix 1 that lines the pore. And this helix interacts with transmembrane helix 3, whereas transmembrane helix 4 and transmembrane helix 2, together with the long C-terminal helix, mediate the interactions between the neighboring subunits in the channel. What comes to CAM6, so as I mentioned, for this protein we were also able to build the atomic models, and in general the architecture of CAM6 is very similar to that of CAM4. We don't really see too much differences in just a little bit in the dimensions, but actually while inspecting more into the pores, so the region of the channels that interest us the most, we could see a very significant difference in the architecture of those pores. And here you can see the comparison of how the pore is formed in case of CAM4 channels and of CAM6 channels. And here you can see purple color or green, those transmembrane helices 1 that lines the pore, that in case of CAM4 are placed vertically so that they form the huge so-called cylindrical pore. Whereas in case of CAM6, we can see that this transmembrane helix 1 is lifted at around 40 degrees in comparison to this helix for CAM4, and it forms the pore of the conical shape. And we believe that those two conformations of transmembrane helix 1 display two different conformations, two different states that CAM channel can adopt. And here one can propose a very intuitive model of how such CAM channels could work, which is the conformation of the transmembrane helix 1 that we see for CAM6 would represent the closed state as it is lifted, and the transmembrane and the conformation that we observe for CAM4 would represent the open CAM channel. However, by inspecting the pores even more, we notice something very interesting in something very unusual. So usually ion channels, like I mentioned already, they conduct charged atoms, so their pores also are lined by the amino acids that are charged. However, what we observed in case of CAM4 is that the amino acid that lines the pore of CAM4 channels are actually hydrophobic. Moreover, by inspecting the densities inside the pore for CAM4 channels, we had found some extra density that didn't really come from the proteins. It was amorphous density that based on also how the pore is built, so like I mentioned that it's hydrophobic, we attributed to the lipids. And we proposed another hypothetical gating models for CAM4, which would be that it's actually the conformation of the transmembrane helix 1 from CAM4 that represents the closed state of the channel, but the channel is not really closed via the protein parts, but via the lipids that reside in that pore. And upon the lifting of the transmembrane helix 1 that we can observe in CAM6 protein, such lipids would be pushed away, and the pore smaller but still free to conduct molecules would be formed. And this was pretty controversial even for us. Nevertheless, we shared this story with research community, and actually not only us, because it turned out that plenty of groups also decided to study the structure of those CAM channels, and they provided the structures for other CAMs, and for majority of those channels, they observed something similar, so extra density that they also attributed to the lipids. So now this concept is pretty well established, but it's still a concept that requires a lot of investigation. But like I mentioned, a lot of groups, including ours, still works on the CAM channels. So I expect that soon the next large step in the field will be taken. And last but not least, I would like to thank some people. So my biggest thanks to my supervisor, Raimund Dussler, that is of a great help during the whole course of my PhD. To Christiane Albrecht, that agreed to us to help us to study CAM channels in context of the placenta, to Jonas Zauk and Marta Sawicka that together with me work on those projects, and also to all the other Dussler Lab members that create really fantastic atmosphere to work in. Thank you.
Thank you very much. Fascinating. Questions, please. Yes.
That was indeed a beautiful story. Yeah, it's really amazing. Do you think that when the last part of the talk talking about moving the lipids to the sites, you actually think that the ATP is required to actually move them? So meaning that the channel is not only moving the ATP through, but also ATP as it's going to be through is also used to move the lipids on the sites.
No, I would rather expect that this change in the conformation of this helix would push the lipids away, because this I didn't mention, but actually between those subunits that form those channels, we could also observe some extra densities that don't really come from the proteins. And we believe that those densities could come from the lipids. So we simply believe that maybe this would be the lateral movements between those subunits, that this could be the pathway. But no, it wouldn't require any ATP. So like the conformational change doesn't necessarily require ATP. It would just require some trigger, some stimuli, or another condition, like for example the change in the voltage of the membrane that they reside, so that it would be more preferable for them to confer this other conformation.
Yes, we have one more question.
Thanks a lot. Can I have questions quickly? So first one, I was wondering how similar are actually the channels within the family? Like, because you mentioned that clearly they have different functions, and then they're expressed in different tissues and whatever. So how similar are they within the family, and is there enough difference or enough similarity to draw a conclusion about the whole family from one structure, or do you think they're specific to perform some sort of ATP transfer in different tissues depending on the class?
Yeah, so like I mentioned, basically now we have the structures for basically all the CAMs, and the general architecture of the channel is basically the same. What people really see the difference in is that for some of them they could indeed see this extra density that all of us, basically all the groups, attribute to the lipids, whereas for some of them, like for example for CAM1 or us for CAM6, we don't really see such density. So it points out that there might be some, I mean, for sure there are some differences, because that's the reason why we have those proteins, that they are different. But I think that the main differences are not really in their architecture. Oh, I didn't also mention that they differ in the oligomeric state. So CALM1 channels are, for example, smaller. So probably they are also there to conduct some smaller molecules, and maybe CALM4 channels could accommodate even some larger molecules like small proteins, not only ATP. But yeah, so I think that the main difference is not really in the architecture and the structure, but rather it will be in the stimuli, the trigger opening of those channels, and this is still, unfortunately, the huge mystery.
Okay, cool. And quickly, second one, because you showed that some of them have different oligomeric state, right? You show the 10 and 11 mers. So which one is it, or is it both, or?
So this we don't know. So those are the results of the overexpression, and to really know how it is at the physiological level. So this would require to purify the native protein from the native sources. It's actually doable. Okay. Okay.
Yes, thank you very much. We have to move
Thank
on,
you.
unfortunately. Talk after. Are you ready for the final speaker of this session? The answer is we are born ready. Okay? Cool. Marcin Drong earned his PhD degree in organic and bioorganic chemistry from Department of Chemistry at Wrocław University of Science and Technology. Wrocław Universities keep on popping. And his thesis was actually awarded the best thesis in organic chemistry by Polish Chemical Society and then Sigma Aldrich, which is a huge company in chemistry. Then he worked in different research institutions in Poland and the US. And 2016, Marcin received professor title in chemistry, and from January 2020, he's heading the Department of Chemical Biology and Bioimaging at Wrocław University. Yesterday, during a meeting in the embassy, he was described by his collaborator as the best chemist in Poland. So I could continue for a long time to talk about him, but I only have about 100 words for this introduction. So I think we should just hear about his fascinating story related to COVID-19.
Yeah.
Thank you very much.
Yeah, good morning everyone, and thank you very much for inviting me here. It's very nice to come again to Copenhagen, although I used to come here before to Novo Nordisk and first time to academic place. So I will basically give you the overview of the technology which we use in my lab, but I will use as an example, of course, COVID-19, because COVID-19 is really very close to what we are doing now, and we've done a lot since really the beginning of the COVID, and I will tell you the whole story basically. So my group is working on proteases. So proteases are enzymes, and we have in humans around 650 proteases. And proteases, basically, they degrade peptide bond. So if you have peptide bond, basically proteases, they can do anything you can imagine. And they are good guys, so basically all these 650 are playing good role, but also they can be bad guys and they can play against you. And we are in my lab focusing on the bad ones, and we are investigating them. So what we are mainly doing, we focus on the so-called substrate specificity. So all the enzymes, basically, they have binding packets where they bind these substrates. And in my lab, basically, we find keys to the logs. So if you imagine as a log the enzyme, so we're looking for the perfect keys. So all these packets, they have different shapes and different properties. So we're looking for the best amino acids which can bind to the certain packets. And we use different types of the technologies, and basically the major technology what we are doing is so-called the combinatorial substrate libraries, and we use fluorogenic substrate, but we also from time to time use the Fred type of the libraries. So What is the specialty of my lab? It's actually unnatural amino acids. So all you know that in humans we have basically 20 natural amino acids, if we consider now those which are proteinogenic. And then it's like my idea back in States was that what about we use unnatural amino acids? At that time it was very much criticized by biologists, also by chemists, that it makes no sense to use the unnatural amino acids because they are extremely toxic and they cannot be used in the drugs, they cannot be used in the crops. So basically they cannot be used in the biological systems. But I decided to focus basically on unnatural amino acids and what are they if we look at the structure. So here you have the phenylalanine, and if you cut this methylene group, then you have phenylglycine, so this is a natural amino acid. If you basically add one methylene group, then you have the homophenylalanine. And here is the first example. So Amgen, a couple of years ago, they introduced a drug which is called carfilzomib, so it's anti-cancer drug, and it contains actually this unnatural amino acid. So for many years, I was really struggling with many people explaining them, it's like, why unnatural amino acid, they can be used, and then Amgen came with this drug. So now, unnatural amino acids, they're getting more and more popularity. So another thing is about stability, because if you use peptides as drugs with natural amino acids, they are very likely to be degraded. And we have many examples of the peptides really beautifully working in the in vitro system, then you put them in the place where they have to work, and then proteases, they chop them immediately. So the idea was that if we have this giraffe, and you can imagine this giraffe being built of the natural amino acids, all these tigers and lions basically, as you imagine them as proteases, they can basically easily eat this giraffe. So we wanted to do something else, and we wanted to do more stable proteins built of the natural and unnatural amino acids and being really highly stable to the degradation. So that was the big idea behind this. So after coming back from States, I gathered all the technology from Big Pharma, because Big Pharma was already sitting on the combinatorial libraries with the natural amino acids. So as a, let's say, secret project, we decided to work on something what is everything what Big Pharma had at that time, and adding the whole technology with the unnatural amino acids. So me and other 3 PhD students, we were working basically on this project for a couple of years, not telling basically a word what we are doing, and as a result, we published a couple of years ago this library, which is called Hybrid Combinatorial Substrate Library. Hybrid because it contains all the knowledge from the Big Pharma about natural amino acids, and we added basically big part of the technology with unnatural amino acids. And using this technology, you can immediately screen any protease you can imagine. And extremely fast, so you don't need anything. You don't need in silico data, you don't need crystallography data, you don't need biochemical data. If you have new protease, and this is what we are doing, you just send us the protease, even the completely unknown, and we can know everything about this protease within 2-3 days. So basically, and then it's like this way, you just deliver substrates, inhibitors, and also the leading structures to every protease of the interest. So how does it look in the numbers? So if you have the natural amino acids in the library, in the short tetrapeptide, you can have 130,000 combinations, and this seems to be a lot. And it's actually not a lot, because we know from Big Pharma and from other papers that this was just the beginning of looking for the selectivity, and I can tell you that none of the really compounds which contains only natural amino acids was ever used as a drug, of course, in terms of the small molecules. With unnatural amino acids, so if you use 130, and now we are going even up to 200, you have 285 millions, now we are even going to 1 billion combinations. So how to test 1 billion combinations? We have this secret technology and then secret libraries, basically, which looks like this. So we use the special mixture of these peptides. So if you give us the protein, you just send in epinor of the protein. So within really 1 day, we can deliver all the information about this protein, about substrate specificity, biochemistry, everything, and then design the leading structures. So I'm not going to go into the much details. We published a couple of years ago the protocol. It's like a big full protocol on how to build these libraries, how to use this library, and how to apply them in the biological studies. And today I will focus as an example on COVID-19, of course. COVID-19 was for me something what we expected, I can tell you, because I used to work on SARS-1. When I was in States, it was very short after World Trade Center problem, and was very short after the SARS. And I used to work in La Jolla, beautiful place, but next to, of course, Pfizer, Novartis, Merck, and other big companies. And also it's like in great academic surrounding for a couple of years. And we're focusing also on viral proteases, on smallpox and on SARS. So we had some experience with this. And after coming back to Poland, I started to work on other viral proteases like Zika, dengue, West Nile virus, and so on and so on. And it's like a couple of years ago, we had a problem with MERS. The problem started, and we have this problem still continuing on. And we're expecting that actually with SARS and MERS, we can have something what will be new. So, and I was not really very much surprised that actually at the end of 2019, I got first gossips from my friends working on viruses that we will have another big problem, and this problem will be actually the worldwide problem because it transmits super fast from human to human. So if you look for COVID-19, basically, in terms of the proteases, then you have 6 proteases, 2 viral proteases and 6 human proteases. So there are 2 viral proteases, MPRO, main protease, and PLPRO, papain-like protease. So those are the viral proteases, and I will focus today on them. But then it's like all of you probably heard about the ACE2, angiotensin converting enzyme. This is also protease, and this is the receptor for the coronavirus. And TMPRSS2, this is the serine protease, which actually cleaves spike. So if you have spike, when spike binds to the ACE2, it needs to be further cleaved by the TMPRSS2, and then TMPRSS2 change the conformation, and then helps the virus to get in. And then we have cathepsin B and cathepsin L inside, which are playing against us because they help the virus to be released and start the replication. So we have all these 6 proteases, and I used to work actually before on cathepsins B and L. I used to work on ACE2, and I used to work on the SARS-PL-Pro from the previous SARS coronavirus. So why these proteases are so important? So if you have the virus getting inside the cell, then it releases RNA, and then RNA it's translated into the polyproteins, basically using the human machinery. And within this human machinery, you have the formation of big polyprotein, and this polyprotein needs to be chopped into the small fragments, and then the small fragments, they are used by the virus to build the new copies, and then starts the replication. So if you inhibit MPRO and PLPRO, these 2 proteases, which chops the polyprotein, you immediately have the great drug candidate, and we knew this from the beginning. So that was nothing new, and we are just waiting for the Chinese to publish the genome, and when they published the genome on the beginning of the 2020, basically we immediately knew that we have 2 really interesting targets without even going into the details. So we decided to focus on MPRO and PLPRO, and I was contacted at that time already by several labs from academia and also from industry, that what about we try to compare MPRO, main protease from the previous virus and from our new virus, and also the PLPRO from both viruses. So I will focus first on the SARS coronavirus MPRO. So MPRO looks like a heart, and it is actually a heart of the coronavirus. So this is the crystal structure, and this is the main protease. So if you have this big polyprotein, it starts chopping this protein, and of course forming the new copies. So here's my good friend Rolf Hilgenfeld. So Rolf, basically, we collaborate with Rolf already for many years. He's a German crystallographer, a virologist, and he is very well known for SARS-1 because he helped to actually quiet down the whole problem with the SARS-1. And for what even he has a sculpture in the Singapore. So we were talking with Rolf already in 2019 at the end that we'll have this problem, but the story started years ago when Rolf was coming to Wrocław and we were discussing about the problem with the coronaviruses, and he was always saying, we will have another coronavirus, we don't have drug, we don't have the test, we have nothing, we are totally not prepared. Pharma is not interested because they neglected previous SARS, although they had the leading structures. So he convinced me to develop one of the libraries focused only on the coronaviruses. And we had this library, we're super lucky, we had this library ready at the end of 2019, and he was supposed to bring me SARS-1 and MERS, MPRO, for testing, and then we had the epidemic at that time. So this is how the library looks like, and the specific point here is the glutamine in P1 position, where you don't have the human proteases with this specificity at the P1 position. So we were hoping that we will use this library for something else, not only for the SARS-1 and for MERS, and then we're super lucky with this information, and then Rolf was actually asked to go to China and test some of the drugs in the beginning of the January, and then he promised me that after he expressed the MPRO, he will do the crystal structure, submit the paper, and in person he will bring the protease to Wrocław, not sharing it with the other labs. And then basically we were lucky because he got very fast the crystal structure, and in person he came to Wrocław, and we started to work in February 2019 on this library, and we wanted to basically get as much information for in silico, for drug retargeting, for design of de novo drugs from this library. So immediately we started screen old MPRO from SARS-1 and the new MPRO. And as you can see here, the more red, then the more active is amino acid in the certain position. And we had this third leucine as being the best. And I decided to release this data for free. So I decided that we will not patent this data. I had no money for this project, and I paid from my own money basically to build this library because no one was interested in coronaviruses. So we released this for free in hope that someone will use basically the structure to maybe design the new drug. And to our surprise, in March this year, Pfizer released a molecule. So the molecule basically, this is the glutamine, which is obvious. This is from the old drug they were using. And then in P3 position, they have our amino acid. This is the third leucine here. So basically what Pfizer is testing now as in phase 3 as the most promising drug for COVID, it contains our amino acid from the screening release early last year. So also we're following this. We got permission to work at the university, and we released all the data about the fluorogenic substrates to help people to work on the drug retargeting. We had commercializations immediately, and Peptide Institute, they signed contract to sell and synthesize these molecules for 10 years, and we have, of course, the percentage from this, from they sell also our inhibitor, but we wanted also to do something from our side based on the data we had, and we synthesized the inhibitor. So you can see this third leucine in this position. We had this way long before Pfizer started to design the molecule. So we designed a couple of inhibitors and also activity-based probes. We tested them. They were very nicely labeling MPRO. And then it's like people, they didn't want to believe us that we have the molecule which binds specifically to MPRO. Then Rolf solved the crystal structure of our inhibitor with the SARS coronavirus 2 MPRO protease. So we could actually confirm that our unnatural amino acids are binding very nicely. And we wanted to use activity-based probes with the fluorogenic group for the imaging of the people with the COVID positive effect. So we actually used our probe for the MPRO imaging. So you basically take the swab and then you add the probe. And we started imaging using confocal microscopy. So you can see here activity of MPRO, main protease in the human cells infected by the coronavirus. Here is the positive example, confirmed, of course, by the PCR, and here you have the negative example, PCR negative. And, of course, we got many offers to publish papers in top journals. We decided to go with Nature Chemical Biology. And Nature Chemical Biology reviewers, they wanted to also us to prove that our inhibitor is working very well. And so we contacted Johan Nates from Leuven, who is a virologist and my good friend. So he has really perfect lab for testing coronaviruses. So they were at that time developing the assay to test different types of the protease drugs. And they tested our inhibitor, and it turns out that this is a very good inhibitor of the SARS coronavirus replication and close to the activity of the remdesivir, which at that time was like a model drug. But in our case, the cytotoxicity was really very low, while in case of the remdesivir, they had quite high cytotoxicity. So with this information in the hand, basically, we are now doing lots of other studies using this inhibitor, and it's also on sale by this Japanese company. We released this data, as I mentioned, for free first. And this paper was assessed more than 130,000 times in a very short amount of time. and used really by many different academic institutions and by pharma to design de novo molecules. So this was the first protease, and now I will focus on the second protease, which to me is even more nasty. So we wanted to catch basically two birds at the same time. So you knew that at that time, the labs were not really eager to collaborate because each one wanted to have the freshest data, the best data. So I was like, okay, with this technology, we are faster than anyone else in the world. So what about we also try to do the second protease in full characteristics? So the problem was that this protease, it's much more nasty because it also, besides cleaving this big viral protein, it also chops off human proteases which are used in the signaling pathway. So basically, if you have the PL-Pro active, the PL-Pro chops off the ubiquitin ISG15, so it stops the cell from sending the signal outside. So we think that this is one of the problems for this virus to being so effective. So this is actually in this protease. So here is Mickey Bekesh. Mickey was PhD student in the States at that time when I was a postdoc, and he expressed PL-Pro1 for me. Now he's working for Big Pharma. So, and he couldn't work because they were in lockdown. So, but he was in Tony Huang lab in New York, where he was working on SARS-1 for a long time after he left Burnham. But also Tony Huang lab was closed. So he talked to Sean Olsen, and basically we built immediately the collaboration in hope that we will get the PL-Pro from States to our lab and we can test it. And then basically we started also collaboration with other groups, and we had many groups contacting us from all over the world saying, oh, we have PL-Pro. So I told them, okay, I will start collaborate with the group who will deliver me active protease. Because the problem was that we got 2 proteases preps from other labs, and they were completely inactive. Also Sean wasn't happy, and I will tell you why, about the activity of this protease. And of course, we used the library, which we just developed also before the pandemic, to work on MERS, because I was looking for money to work on coronaviruses. Yeah, so we had no chance to work on them. So we used this library. Immediately, we screened the protease. We did show that there is completely no mutation in the active site in the PL-Pro, which is great for the drug design. Then we tested different types of the substrates, and these substrates, they were extremely selective for the viral protease. We developed inhibitors in hope that we will get as first crystal structure, because there was a race who will get first crystal structure of the PL-Pro. And I had some experience with this. So you know how hard it is to get the crystal structure of the protein with the inhibitor, but we sent the inhibitors to Sham, and within a week we solved 2 crystal structures. And those crystal structures, they were for a long time first crystal structure of the PL-Pro with the small molecule drug candidates. And we decided to look for the activity, because one of the things was that we knew that the SARS-1 PL-Pro, it's active on ubiquitin, and then it's like this PL-Pro from SARS-2 wasn't that much active and was really extremely active on ISG15. So that was something new for us, so we're suspecting that there is a lot of mutation on the surface, and these mutations will actually change the specificity and make this protease even worse comparing to SARS-1. So we did the crystal structure, we compare it with all the mutations, and indeed, in active site there is no mutation at all, which is great for the drug design, but on the surface there is a lot of different mutations. And for a year, from that time, we decided to focus on various variants of the coronavirus. And here is one example, because now we are talking that we have mutation in the spike. Spike is not the only protein which is important for coronavirus being so bad between the variants, but also PL-Pro. So we found the mutation in PL-Pro, which makes PL-Pro in the version gamma much worse comparing to the, let's say, the original coronavirus 2 version. We just published this paper also, and it also shows that designing the drugs which binds outside of the active site makes no sense. We release all this data also for free, and here is something what we are doing now. Just 2 slides. We are working on drug retargeting. We have lots of projects, lots of collaborations, lots of new drugs in testing. One of the most promising is epsilon, which inhibits very nicely both of the proteases. So we have a paper submitted about this. Also, we work also on other inhibitors, which I cannot tell you now, because we have lots of the agreements, of course, with the groups we collaborate on coronavirus. So in summary, both proteases are excellent drug targets. Whichever you inhibit, yeah, you immediately kill the virus. The binding sites are identical with SARS-1, which is great for the drug design. And we need a drug for the next coronaviruses because vaccine will not work. And if we have a drug, we have a high chance that basically we have the new coronavirus, then we will have a drug which will help to kill the virus replication immediately. And of course, thanking Viola, who was my PhD student and who I convinced to work on this project. So she scooped 4 papers, first author papers in top journals within a year, working basically on this project. So Viola is really great, working on viral proteases. And of course, lots of new collaborations. Kasia, who was my PhD student, now she's a postdoc in Vishva Dixit Lab in Genentech. Miki, who is working on this project, my PhD student, but also groups, Scott from SBP, where I was a postdoc, group of Miki and Tony in New York, ending up.
Thank you.
And all the others. And thank you very much.
Thank you. Thank you. I think it's going to be another invitation to Copenhagen for more time, for a bigger lecture one day.
Yeah. Oh, we have a lot. Yeah, maybe I go to.
Thank you very much. One question. Who goes first? Okay. That was good.
It was fascinating talk. I'm just, I guess you have a lot of cool stories to share, in the background of all this collaboration and so on. But short question about delivery. So what do you envision how this drug or inhibitor should be delivered in this way?
Okay, so I can tell you the whole story basically looking for what Pfizer was doing, because Pfizer first, last year, they released the information that they are going to test their first inhibitor for MPRO, and they started clinical trials a year ago, basically in September. But this was the molecule which they developed for SARS-1, and knowing that the active sites are exactly the same for SARS-1 and SARS-2, they decided to test this molecule, but it was intravenous. And then the intravenous basically was a problem with toxicity, and to my knowledge, now they stopped this project. And then they started to do de novo design of the inhibitor based on all this information they can gather. And now the idea is that this drug is given orally to the people. So, and it works, and now they are actually in the second and third phase of the clinical trials. So I would say these drugs will be given orally, and this is one of the things with unnatural amino acids, because this molecule, their molecule is built totally of the unnatural amino acids. They are super stable. So you can even eat this drug, and then it's not being degraded by the human proteases in the digestive tract, gets very nicely absorbed. And also there are some tests with the inhalation that you can spray this drug, and then because the virus is usually on the surface, and then do this type of the therapy too.
Thank you very much. Please, can I please have all the speakers from this session here with me? Thank you very much. One more time.
Oh, thank you.
One more. Something sweet. Here you go. Now it's time for lunch. We will meet 20 to 3 right here and continue with our final session of the day. Okay, so please enjoy.
Thank you.